John wrote into our Facebook page, "Ian, do a show on the 2q13 fusion site. This argument for fusion is so lame it's unbelievable." John is referring to the alleged biological fusion of 2 chromosomes in humans. To shed some light on this wide-ranging topic, I am joined by Dr. Jeffrey Tomkins via Skype from his office in Dallas. Dr. Tomkins has a PhD in genetics, as well as a master's in plant science and a bachelor's in agriculture. While working as a faculty member in the Department of Genetics and Biochemistry at Clemson University for over a decade, he performed genomics research in plants, animals, and microbial genomes. He is now a research scientist at ICR. He has 56 publications in peer-reviewed scientific journals and 7 book chapters in scientific books. Since coming to ICR, he has 15 publications in peer-reviewed creation science journals. He is the primary author of The Design and Complexity of the Cell. Welcome to the show, Dr. Tomkins.
Thank you. It's great to be on.
Now, your expertise is in genetics, and whenever I have given a talk, I like to poll the audience and ask how many people have heard that humans and chimps are 98.5% identical genetically. And invariably, it's a lot of people, usually about half the audience, and these are just lay people. So obviously, the propaganda machine has been working like a well-oiled machine. Now, you conducted research with Dr. Jerry Bergman into the published scientific literature. What did you actually find that contradicted this paradigm?
Well, that's where we first started, or I first started, and I linked up with Jerry Bergman, a biologist and biochemist. And we found out that by studying the scientific literature that these 98% claims are based on very small fragments of the genome that are compared. And in fact, with my background in bioinformatics, I could go into these papers, look at the materials and methods, and see where they threw out sequence, or what sequence was it, thrown out or omitted by the algorithm. Because there are certain sections of the genome that just don't line up. There are sections of the genome that are present in humans, absent in chimps, present in chimps, absent in humans, or there's large gaps in the sequence that the algorithm doesn't really go through. It stops. And so I was able to look at all these regions of the genome that all these different papers looked at. And we found out that it varied, but when you threw back in the DNA sequence that was tossed out, that the average similarity for a lot of these papers varied, depending on the paper, varied between 66% all the way up to 86% similarity. And so what you're getting really is just this cherry-picked data. You're not getting all the DNA sequence added in that should have been added in to give you an overall similarity. And another thing that these papers did was they only looked at, a lot of times, just what are called exon sequences or protein coding sequences in the genome, which is, in humans, it's about 2% of the entire genome. And also they would only look at the protein coding sequences that were present in both genomes. And actually what I've found out, and we'll talk about maybe a little later, is that close to 7% of the protein coding sequences in the human genome are totally missing in chimpanzees. So there's a lot of there's a lot of bias going into these papers. They're really just comparing small fragments of the genome that are highly similar. And even the evolutionists now are backing off of this in the scientific literature, even though you still see this myth persisting in the common media or the popular media.
You have also done your own bioinformatics research comparing the chimpanzee genome to the human. What did you find?
Well, one thing I had to do was overcome the limitations of the common algorithm. It's called BLASTN, that's used to compare DNA sequences. It's probably the most commonly used algorithm. So the BLASTN algorithm will stop when the sequences become too dissimilar. And so that's what's going on with chimpanzee and human. So I had to basically slice the chimpanzee genome up into very small pieces and then compare those fragments. And I had to also do that, for multiple data sets. I had to find the optimum sequence slice, or the optimum fragment size that would line up optimally to human. So I did that for every single chimpanzee chromosome. And of course it varied depending on the chromosome, but overall the genomes are only about 70% similar. So the chimpanzee genome was only about 70% similar compared to human. And this actually matches up with a lot of evolutionary papers and their data, because when the chimpanzee genome first started to be sequenced, a paper was published in 2002, and they found that only about 2/3 of the sequences, the raw what they call the trace reads, or the reads off the sequencer after they were cleaned up, only about 2/3 of those would align to human, which is about 70%. So I was actually seeing what had been published earlier in 2002 and in subsequent papers. So this isn't like it's far outdated or anything. It actually confirmed what was previously known but not really popularly discussed in the media.
Oh, isn't that interesting?
So the chimpanzee, on average, the chimpanzee genome is only about 70% similar to human. And this is the study I did was probably the most painstaking bioinformatics study that's ever been done on the subject. And it really just confirmed what was already known anyways, but not really talked about.
So your study covered the entire genome and not just that 2% of protein coding DNA. Is that correct?
That's correct. I studied the entire chimpanzee genome, chromosome by chromosome. And I'm actually doing the same thing right now. I haven't published this data yet, but now I'm doing the other side of that comparison, because that's just asymmetric. You're comparing chimp against human. Now I'm actually comparing human against chimpanzee. And I'm not only just comparing every single human chromosome, but I'm also looking at the coding regions. I'm looking at what are called the promoter regions or the control regions in front of genes. And I'm also doing this very systematically, so we can really see what regions of the genome are the most similar, how similar they are. And so when I get done with this current study, it's going to sort of be the end-all genome-wide comparisons, but I don't think it will really tell us a whole lot.
That should prove an interesting study.
No, it'll just kind of confirm things.
So, for the sake of viewers, you mentioned comparing chimp to human, and now you're going to compare human to chimp. The reason being because chimps have orphan genes that humans don't have, and vice versa. Humans have genes that chimps don't have.
And vice versa, right.
Which is the reason why you need to compare it both ways.
Right. And of course, the chimp genome is actually biased, because it is and still is being assembled based on the human genome as a framework. Because in DNA sequencing, you can only sequence little pieces of DNA at a time, and then you have to assemble those sequences into contiguous sets of overlapping sequence, and eventually you hope to actually reconstruct entire chromosomes. And so they have assembled the chimp genome, not only the chimpanzee genome, but the orangutan genome and the gorilla genomes and the macaque genomes based on the human genome. And they're still doing this. There's a paper published in 2013, and I looked at the materials and methods, which actually weren't in the paper. You had to dig somewhere else for those. But they were actually assembling all these ape genomes based on the human genome, and not just any human genome, but the 2006 version of the human genome. Which is what? It's 2014 now, right? So this was an 8-year-old version of the human genome, but they had to be consistent because the original version of the chimpanzee genome in 2005 used essentially this version of it. It's called HG18. We're now on what's called version HG19.
Now, tied in with that, the anti-creationists have loudly touted what they call chromosomal fusion as proof positive that humans and chimps shared a common ancestor. So basically, our DNA is wrapped up in these bundles called chromosomes. We have 46 pairs of chromosomes, whereas the chimpanzees and the great apes have 48. And so because we were supposed to have branched off from an ape-like creature, the evolutionists have proposed that 2 chromosomes from our ancient ape-like ancestor fused together when we humans evolved. Dr. Kenneth Miller touted this. I want to read this quote. This is what he said at a lecture given at Case Western University back in 2006. In fact, Miller was very much gloating over his recent victory against the intelligent design camp, and this chromosomal fusion was one of his primary weapons. Quote, "What must have happened is that one pair of chromosomes must have gotten fused. So we should be able to look at our genome and discover that one of our chromosomes resulted from the fusion of two primate chromosomes. So we should be able to look around our genome. You know what? If we don't find it, evolution is wrong. We don't share a common ancestor. End quote. He then went on to claim that recent research had found this fusion site exactly where evolution predicted it would be found, on chromosome number 2. What have you to say on this?
Basically, humans have 48 chromosomes, apes have 46. So based on what are called banding patterns of chromosomes, which are observed under a light microscope, and these are actually condensed chromosomes during a certain part of the cell cycle, not what you would normally find during a living cell that's actually functioning in your body. But in these condensed chromosomes, they stained them and then looked at the patterns in the chromosomes under a light microscope. And there were some banding patterns that were similar between some human and ape chromosomes. So they thought these smaller ape chromosomes fused to form human chromosome 2. Well, then this was in 1982. Then in 1991, they actually found what they thought was a fusion site, but it wasn't what they were expecting. It was a different pattern than what they have found in living mammals that had actually undergone a chromosome fusion. It did not contain something called satellite DNA. So in living mammalian fusions, they find satellite DNA combined with satellite DNA or satellite DNA combined with telomere DNA, but they never find telomere-telomere fusions. It's never been heard of. So if it actually did happen in humans, it would be the first such case. And anyways, this fusion site consisted of these telomere sequences, but it was very small. Human telomeres contain 10,000 to 15,000 bases of this sequence. So in other words, if you were to find such a fusion, you would expect something about 20,000 to 30,000 bases in length. But this fusion site was only about 800 bases in length, and it was only about 70% similar to what would be a pristine fusion. Yeah. So it was highly degraded.
Because they found these alleged fusion sites all over the place in other chromosomes, right?
Well, it turns out that internal telomere sequences are found all over the human genome. And there's actually been papers published on this in the past few years. And it turns out many of these sites are performing functional things in the genome. They're binding to things called transcription factors, which initiate a gene or cause a gene to begin to be copied into RNA. And as it turns out, in my research, the fusion site is actually read in what's called the reverse complement, and it's a transcription factor binding site in the middle of a gene called DDX11L2, which is an RNA helicase long non-coding RNA gene. So it doesn't code for a protein, it codes for a functional RNA molecule that's active in the genome. And it turns out this gene is highly co-expressed with other genes that are highly critical to the cell cycle and signaling in what's called the extracellular matrix, or the area outside your cells where a lot of signaling and communications go on. And it turns out this gene is highly expressed in many different cells in the human body. So it's a very important gene, and it's linked to many other highly important genes as well.
So this alleged fusion site is in the middle of this life-supporting gene.
Right.
The gene crosses over the fusion site.
Yes, across it. Actually, this site is in the middle of the gene. It's in what's called the first intron. So these genes, these long non-coding RNA genes, they function exactly like protein coding genes. They have exons or coding regions that end up in the final RNA molecule. They have introns, which are intervening regions that do not end up in the final RNA molecule. They have promoter regions and control regions exactly like protein coding genes. And in fact, your genome contains over twice as many long non-coding RNA genes as it does protein coding genes.
Wow.
And they're finding out now that these things are critical for human health, growth, development. They're finding out that when many of these long non-coding RNA genes are mutated, that it leads to cancer, that it leads to developmental problems. And so this is actually the hottest area of research right now at the NIH, and their funding mechanisms is these long non-coding RNA genes.
So in our alleged ape ancestor, this chromosome would break into two, breaking this critical gene in two, so the ape couldn't live.
Well, what I'm saying is that you don't form new, highly functional critical genes by slamming together two chromosomes. And
yes, that's a much better way to put it.
And what's really interesting is that there was a research group in 2002 that sequenced 600,000 bases of DNA surrounding this gene. And they actually reported that the fusion site was in what they called a pseudogene at the time, which is the gene I'm talking about now, which we now know 12 years later is highly functional and critical to the human genome. But they actually reported this back in 2002, but no one was really talking about it. But they sequenced 600,000 bases of DNA surrounding this gene and found that it was in a gene-rich region. It was in a nest of other genes, and that none of these genes in this entire 600,000 bases are found in chimpanzee.
Now that's a crucial
So
point.
they hypothesized, yes, so that, and of course my research showed that as well, but they hypothesized that all these genes got there by coming from other places in the human genome. They didn't come from chimpanzee. This is the evolutionary thinking, the mindset. They didn't come from chimpanzee because there was no comparison to make. So they just came from other places in the human genome. So they just somehow ended up there through different mechanisms, supposedly. But
isn't that a convenient story?
yes, right. It's not one thing; it's another.
So, what would happen to me if this gene were removed?
Well, it's difficult to study because this gene is not in primates. It's not in mice. And see, these are our model systems, especially the mouse model system is very important to studying human health and disease, because if that same gene is in mouse, then we can knock out that gene and see what the effects are. But you can't really do that in humans, or at least talk about it if you did. But anyways, when we study human genes, we have to do it in a system like mouse, where we can knock out the gene and mess it up and then see what happens. And so there's a lot of genes in humans that are not shared by other creatures, especially lab study creatures, and we just really don't know what they do. We can associate those with disease. We can associate those with the expression of other genes and see what they're linked to in the genome. And so that's what I did. I went into the databases and deciphered what other genes this gene was linked to, what other genes it was co-expressed with. And this is very solid data, actually, because it shows that this gene is linked to the expression of other genes, and it's very important. And in fact, this gene was linked to genes that are at the top of the pyramid of many important processes in the cell.
Now, after I discussed this paper on the show, we had one gentleman write in. He was very cordial, and I appreciate that, but he was skeptical of your work. Enrique's argument was that the gene encompassing the fusion site did not produce any known protein.
Well, my response to that is that we now know that there's over twice as many non-protein coding genes in the genome, which I just talked about, called long non-coding RNAs, that are highly important to the function of the genome. They're just as important as protein coding genes. And so his comments are made out of complete and total ignorance as to what and how the genome functions, because we now know that long non-coding RNAs and non-coding RNAs are the key to understanding genome function. It's like the protein coding genes are building blocks, and so these tend to be more similar among different creatures. But the non-coding parts of the genome that produce these non-coding RNAs are actually more organism specific, or what they call taxonomically restricted. And it's like these genes provide the metadata for the building blocks that are the protein coding genes to function. And they do a lot of different things. These non-coding RNA genes are found in the nucleus. The products of them, the RNA products of them, are found in the nucleus. They're found in the cytoplasm. Some of them are even exported outside the cell to other cells in structures called exosomes. So these non-coding RNAs do a whole plethora of things depending on the gene. And they're highly critical to the function of the cell. And like I said, this is the most important area of research right now in biomedical genomics that's going on, and it's receiving the most funding, and it's part of the ENCODE project, what we call the ENCODE project.
So they are critical life-supporting genes.
Yeah, they have knocked out these long non-coding RNA genes. If they have similar counterparts in mice, they have knocked them out and actually found that many are lethal, and that they're highly important. And in fact, many of them have been associated with all sorts of different types of cancer in humans.
All right, I know how much the viewers appreciate all this, as do I. So from all of us, thank you for taking the time to be on the show today.
Well, thank you. It was great to be with you.
You can get a copy of Dr. Tomkins' brand spanking new and educational book, The Design and Complexity of the Cell, from ICR. Now, this interview was edited for television, but you can catch the entire interview on our YouTube channel. Just head on over to genesisweek.com, which will take you there. Stick around. We'll be back in just 1 minute.